Okay, this is a recording of the Lightning Round, which I did at GIS 2020 in Orlando. Basically, it's 20 slides, which forward automatically every 15 seconds, so you have about 5 minutes exactly to present your subject. Okay, so here we go. Okay, thank you very much for letting me loose over here. I'm John Dempsey, a greenkeeper and sometimes scientist from Ireland, and I'm talking about Microdochium nivale infection. So, what exactly am I talking about? Well, it's known as Fusarium patch or pink snow mould, and it affects most cool season grasses, predominantly during cool, moist conditions and under snow cover. It's an Ascomycete fungus, and it's the most common and damaging pathogen of turfgrass in the United Kingdom and Ireland, and a very important pathogen in the northern areas of the US, Europe, and throughout Canada. So anyway, I carried out an extensive study during my PhD research of the infection process, and I used many pot samples and turfgrass species in the greenhouse, as you can see here. And of course, being in Ireland, I also had no shortage whatsoever of outdoor samples to work with. So here, as you can see, plenty of infected trial plots and greens to choose from. So I had these many samples in the greenhouse and trial areas, and I utilised numerous laboratory techniques, but one in particular was called fluorescence microscopy. So what does this involve? Well, it involved using various dyes which attach to the fungal cell walls, and when viewed under ultraviolet light using a fluorescence microscope, the light of the fungal structures became very clear. And you can see that in these photos here of the various infected leaves under different parts of UV light. This is a very useful technique. So what we found was that, as you can see in the examples here, the top photos show various stages of the infection, and the UV light really helps to focus on and observe what was going on. We also use a similar technique, as you can see in these photos on the bottom, to look at various defense responses in the infected turf grasses. So this is what we found. The source of infection, the inoculum, are Conidia, the reproductive structures, and hyphae. And these we found were present year round in the thatch or root zone. And you can see in these photos here the hyphae, the blue hyphae, in amongst the sand particles. So when conditions were right, environmental conditions, the hyphae would grow from the base of the plant over the crown and sheath and onto the turfgrass leaves. So here you can see a creeping bent leaf covered in a mass of hyphae or mycelium. Microdochium covers the entire leaf. Again, this is another photo showing the mass of mycelium on it. The red is actually chlorophyll, which autofluoresces red under ultraviolet light. So the mycelium is infecting the plant, it's all over the leaf, and with microdochium it actually enters the plant via the openings, the respiratory openings, the stomata. And here you can see the infection hyphae entering the plant via the stomatal penetration. You can see it quite clearly, the blue hyphae entering the circular stoma. So the microdochium enters the plant via this method. Once it's in the plant, it actually then travels through the intercellular spaces into the cells and extracts nutrients. It's just like any other organism. It just wants to eat and drink, and like other organisms, once it's had its fill of eating and drinking, it likes to reproduce. And it does this by exiting the stomata again, producing numerous reproductive structures. These are the Conidia. And you can see them here. Thousands, if not millions, are produced following infection. These exit the plant and they're spread. They're very small, 25 microns is the average length of them. You can see in the bottom photo here on the right, one of these Conidia actually germinating and the hyphae growing from it. So these Conidia are then spread by wind, by rain, and the whole process of infection continues. So hyphae and Conidia are in the soil thatch root zone. They are the main source of inoculum. When environment and conditions are suitable, the infection begins and the mycelium grows from the base of the plant. Infection with Microdochium is via stomatal penetration. It infects the plant, extracting nutrients, emerges from the plant, producing these Conidia, which are the means of propagation and dispersal. But what we should note is infected plants respond with some very complex biochemical defense mechanisms. First line of defense includes what's called the hypersensitive response, and it relies on hydrogen peroxide, which is key for this. And you can see this is the blue here showing at the infection sites. There's then a more systemic or whole plant defense, which involves production of a whole range of defense compounds, mostly polyphenolic compounds, and we studied these. Here what we can see is the level of these defense polyphenol compounds. The blue are the level of these compounds in uninfected plants, and the red shows the elevated levels in infected. This is over 3 years with Poa annua. What we also did then was we treated turf grasses sequentially with phosphate and found that following 6 months of these sequential treatments, these defense compounds were much elevated in the treated plants, as you can see in the red compared to the green and blue. So these defense compounds were elevated also in our infected plants. These are greenhouse samples. The phosphate pre-treated plants had much higher levels of defense compounds upon infection, and subsequently much lower levels of disease incidence. So that was it. That was the 5 minutes of our lightning round, and hopefully you enjoyed it. As I say, 5 minutes, 15 seconds per slide, and that should be on the way out. Thank you very much.